All right, hi guys. So this lecture goes along with actually no notes. So there are no notes that go along with this. Instead, today we're going to be learning about dihybrid crosses. And you should have a worksheet that looks something like this. It may be a different color, but at the very top it says dihybrid cross practice, and you will see your first really huge dihybrid cross that looks like this. So make sure that you have this packet in front of you. We're going to do a couple practices together that you'll just kind of watch here on my screen, and then eventually we'll do two big, huge squares. So we'll have plenty of practice together before I cut you loose and you finish the worksheets. Okay, so this is an example of a dihybrid cross. You can see that there are, instead of just 4 squares in the Punnett square, these dihybrid crosses have 16 squares. I'll also point out right now that these do pop up on the air test. So I have intentionally tried to take my time and make sure that you guys get plenty of practice. So the beauty of a dihybrid cross is that it actually is going to track 2 traits at the same time. So we're going to see the chances if 2 individuals, still again, so if we have these 2 individuals, if we still have 2 parents and they were to have offspring, what are the chances that they'll be, this trait and this trait? What are the chances that they'll have brown hair and blue eyes versus just one individually? So the basic steps is first, you're going to figure out all of the possible sperm and all of the possible egg genotypes. And you'll see what I mean with these steps in just a second. So first, we'll figure out all the possible sperm and egg genotypes. Then we're going to lay those gametes on the top and the side of the dihybrid cross. And then we're going to cross those alleles by just taking time in a very organized fashion and filling in all 16 boxes. And then we're going to look at the cross and try to analyze those results. These crosses can get really busy. So I'll also spend a lot of time today just focusing on making sure that your letters are, they're nice and neat, and you can clearly see if it's a capital or a lowercase allele and so on. So let's look at this first example here. We're going to look at a pea plant. And so we're going to look at 2 traits. We're going to look at both seed shape and seed color. So the alleles here for seed shape, they can either be round, which is dominant, or wrinkled, which is recessive. And then the seed color it can be yellow, which is dominant, or green is recessive. So, and you'll always be given this information. You'll also always be given the male and female genotypes, so the parents. So the male parent genotype is big R, little r, big Y, little y. And then the female happens to be the same. So the female parent is big R, little y, little y. And our first step, like I said, is to figure out all of the possible sperm and egg combinations. Now remember, one of Mendel's laws was that when each parent goes through meiosis and each sperm or egg gets 1 allele, it is totally random. So there's no rhyme or reason to it. So our job here is to figure out all of the possible combinations. So if you look at this male, for example, there are 4 possible combinations of the alleles that he could pass on to the sperm. So one thing, I think you guys have seen this in algebra. Some of the times we'll use FOIL. So with this, FOIL means first, outer, inner, last. So we're going to do the first of every letter. So if you look at the male genotype, the first R and the first Y. Okay. So if you look here, I'm going to put, the first R and the first Y are going to go first. So big R, big Y goes here. The next possibility is the outer. Okay. So the outer R and the outer Y. So we get big R, little y, so the outer. Next, the I in the foil means inner, so the inner R, the inner Y. So we'll have a little r, big Y. And then the L stands for last, so the last R and the last Y. And here we have the last sperm here would be little r, little y. Now let me tell you, this foil thing works for me. A lot of you just have this ability that I don't have, and you can actually look at that male genotype and come up with four, all the four different combinations. My brain just doesn't do that. So whatever works for you, but I like foil because it makes sure that you don't miss one combination. So let's go through the female again. Let's go through the female now. So we have her genotype. We're going to do the first, right? So in foil, we're going to do the first R, the first Y, which is big R, big Y. And there we have it. We're going to get our second one, which is the outer. So the outside R here and the outside Y. So big R, little y. There we have it. And then we're going to have the I stands for inner. So the inner R and the inner Y is little r, big Y. And the final is the last R and the last Y. So the L in foil stands for last. So we have little r, little y. Again, what this is representing is this female plant, given her genotype that we were given, when her cells go through meiosis, there's 4 possible combinations that she can pass on to her offspring. And that's what we've just done. So the 4 possible combinations for her are big R, big Y, big R, little y, little r, big Y, and then little r, little y. Once we have the possible combinations for sperm and egg, we're going to plop those on the Punnett square. So actually, if you look at my worksheet, well, actually, I think I have the blank one. Okay, so here we have a blank Punnett square, and we're going to enter the possible sperm. So looking back, if we had it in front of us, the first sperm combination is big R, big Y. So you can see I've entered it there on the side. And then I'm going to quickly just enter the remaining 3. So those are the 4 combinations we just came up with using foil. Then I'm going to plop all of the possible egg combinations here on the top. Okay. Next, we're going to go through and fill in the square and just slowly take our time and carry down all of the letters. So if you look here at the first square, we'll carry down big R, and big Y, big Y. Big R, there we go. And big Y, big Y. Notice a couple things that I've done intentionally to organize all these alleles. Notice that I put my R's together and my Y's together, okay? It's going to be really important when we're looking at all these letters, especially when it's written in your little letters on your paper, that it's organized and clear. And one of the best ways to do that is to make sure you pair up your R's and then pair up your Y's and put the dominant ones first. And I'll show you that in the next one. Let's fill in our next square. So this one, I've carried the big R, and then big Y and a little y. And notice again, I've clumped the letters. So R's are together and my Y's are together. And notice that in this case, because I do have a recessive allele, I put that second. Okay, so I have big Y, little y. If I move on to the next one, again, I've put my R's and Y's, paired them together, and then put the dominant ones first, and so on. So you'll just take your time, slowly go through and carry and fill in the blanks. So eventually the square will look like this, and it looks pretty neat and tidy on this, on the screen here, but I know even my handwriting gets very messy. So just really be intentional about organizing your letters and making sure the bigs and the littles are really obvious, even if you have to do them very strangely different. For example, with my Y's, I do capital Y's like that, and then I make really weird little lowercase y's. Okay, so just be mindful as you go through. And then you're going to analyze it, right? So we said the last step was to then analyze the results, which is really the whole point of filling in the squares. So in this case, we want to know how many individuals would we expect to be round and yellow. Both of those are the dominant trait. So any square that has at least one big R and at least one big Y is what we're looking for. Oops, sorry. Sorry. So for example, this would be round and yellow because there's a big R and a big Y. This would be round and yellow because it has a big R and a big Y. This would be round and yellow. 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 And so far that's 6. This is round and yellow, 7. This is round and yellow, that's 8. Round and yellow is 9. And that's it. So 9 of the offspring would be both round and yellow because they have at least one of the dominant alleles of each. Round and green is going to have a dominant R and then 2 little Y's. Okay, so let's see. Here's one, right? A dominant R and 2 little Y's is round and green. So that's 1. Here's another one, 2. And 3. And that's the last one. And so you'll continue on and you would look, let's do wrinkled and green. So wrinkled and green is both recessive. So all recessive alleles. In this case is just 1, I think, right? So just 1. And then I've entered my answers. So, which is pretty much what we found. So 9, 3, 3, and 1. If at any point you want to go back and watch the whole beginning of this square, you can always go back. And here I like this because this is the same cross we just did, but there's a visualization. So you can see that we actually are tracking 2 traits in this Punnett square, right? So both the color and the shape of the seed are changing depending on their genotype. We have round, we have yellow, we have green, smooth, and wrinkled. Okay. Let's do another one. And in fact, this one is in your packet. So this should be the first one on this worksheet, should match up with what I've put on the presentation here. So let's go through. We're looking at rabbit hair color and rabbit eye color. And you can see that gray is dominant to white and then black is dominant to red. Given the male genotype and given the female genotype. And so our first job here is to figure out the possible sperm and egg. And remember, I'm going to use foil here. So with the male genotype, I'm going to do the first of both letters. So the first G and the first B. Big G, big B. The next one is the outer. So the outer 2, which is big G, little b. The I stands for the inner 2, which is little g, big B. And then finally the last. So the last of the G's and last of the B's, which should be little g, little b. Okay, next one, we're going to do the same for the female and her possible combinations of eggs for the eggs. So we're going to do the first G and first B. So little g, big B. The outer, so the outer G and the little b. The inner, so inner G, sorry, little g, big B, and then the last of both. So the last G and last B. Whoops, sorry. So here we are. This is exactly what it should look like on your worksheet. I just kind of cut and pasted my key. This is exactly what we just did. Okay, so those should be your possible sperm and possible eggs. Again, if you need to review that process, review the last minute and go through it again. So now we're going to plop those on the Punnett square. Okay, so here's my Punnett square, and I went through and plopped exactly what we just did, and put one parent on the side and one parent on the top. Okay. Next is to just fill in the blanks. Do it slowly. Make sure that you're being really neat and tidy with your letters. So you can see here, I've just carried down the alleles, organized my alleles, and I've put the dominant first. Okay, here we go. Same thing. So I have big G, big B, little b. Next one, I still keep my G's in front, right? Because I'm still just organizing both letters and then putting a dominant first if it is there. So I have my G's first and then my B's. And again, okay, still just carrying down these alleles. So this got a big G and a little g, and then big B and a little b. And so on. Just keep going. Take your time. Take your time, keep your letters nice and neat. And so eventually you should have your Punnett square should be completed like this. And if you want to pause and fill in your square, that's what I would do. So at this point, my square is complete, and now it's time to analyze the results. So the questions here are how many rabbits will have gray fur and black eyes? And those are both the dominant traits. So if I pull up my square, again, anything that has at least one big G and one big B is what I'm looking for. So this individual, right? A big G, big B, big G, big B, big G, big B, and so on. So I got a total of 6 that would have gray fur and black eyes. Next one is gray fur and red eyes. So at least dominant G and then both recessive B's, right? So here are my blue circles. So big G and both recessive B's, or there's only 2. Next, how many rabbits will have white fur but black eyes? So in this case, now we're looking for 2 little G's and at least 1 dominant B. And these are represented by the little orange dashed lines or whatever. So little G's and at least 1 big B. And there's 6 of those. And then finally, the white fur and red eye is just all recessive. So all 4 recessive alleles, and there are 2 remaining. And sometimes you guys will be analyzing your Punnett square, and I kind of like to circle like this, and it helps you keep track. Sometimes you'll get to the end and you're like, oh my gosh, I didn't even count that critter. And it kind of makes you realize you need to go back and just quickly check yourself. So you should make sure here at the end that you actually do have 16 accounted for. Okay, again, if you need to go back to that first problem, the first page of the worksheet, please stop, go back, or push pause and finish. But what I'd like to do is I'm going to keep moving for the sake of the video here, and I'm going to do question number 2 with you, and then you're going to do question 3 and 4 alone. So if you look on the worksheet here, now we're looking at pea plants again. So we're going to look at 2 different traits. We're going to look at plant height and flower color. So plants can either be tall or short, and red or white. So the female pea plant is homozygous dominant for plant height and heterozygous for flower color. So we know the genotype homozygous dominant must be big T, and for plant height, and heterozygous for flower color, so big R, little r. So she's tall and red. And then the male was heterozygous for plant height, so we have big T, little t, and heterozygous for flower color, big R, little r. Okay, So here we have those genotypes that we just came up with. Now we're going to first figure out the possible sperm and the possible eggs. And again, I'm going to use foil. So I'm going to quickly, whoops, oh golly, sorry. I'm going to quickly do the foil with the sperm first. So the first T and the first R, the outer T and the outer R, the inner, and then the last T and the last R. I'm going to foil now over here for the female and figure out her possible combinations for the eggs. So the first T and the first R, the outer T and the outer R, the inner T and the inner R, and then the last T and the last R. So here we have our 4 possible combinations for each. And now we're going to plop those on the Punnett square. Sorry, I forgot. Here's, again, exactly what your worksheet should look like, just exactly what we just did. So if you want to check yourself. But I'm going to keep rolling. So now I'm going to plop those on the Punnett square. So you can see I've put 1 parent on the top and 1 parent along the bottom. And I'm going to take my time, keep my letters really neat and tidy, and fill in the squares. Okay, so here's my first square. I've carried down a big T and a big T, and then a big R and a big R. And I'll do the next one, and I get a big T and a big T and a big R and a little R. And I'll just keep moving right along, keep scooting, taking my time, making sure I'm keeping my T's together and my R's together, and so on. Okay, now we're going to analyze the results. So how many plants will be tall with red flowers? Those are the 2 dominant. So how many at least have 1 dominant T and 1 dominant R? So if we look at the Punnett square, there's a lot. Okay, so how many is that? 4, 8, 9, 10, 11, 12. Lots. So in this case, a lot of them are going to have the 2 dominant phenotypes. So 12. How many will be tall with white flowers? So this is the dominant T, but little R's. And there's 4, right? So dominant T and little R, dominant T and little R, and so on. And so then the rest are going to be 0. So sorry, if we go back to the Punnett square, you'll see right off the bat that after analyzing the results, all of them are going to be one of the first 2 options. So the last 2 are 0. Okay, so we've done 3 total together. You should have half of your packet completed. So what I'd like you to do is keep moving and do question 3 and 4 on your own. Remember, the first thing that you're going to do is come up with the possible sperm and egg just using foil. And then you'll enter those, you'll plop them on the top and the side of your Punnett square, fill in your square, taking your time, keeping things organized, and then analyze your results. All right, and that is it for some real dihybrid cross fun.